He's based in Taiwan, and we will go back in time now. We will go to look at probably more fossil fish. But yeah, here we go. Thank you so much for making it actually quite early in Taiwan at the moment. So thanks for making it today. The
Yes.
floor is yours. I can
Thank
see your
you.
screen. Oh, good.
Yeah, thank you for the invitation. My name is Chen Xianglin. I am Assistant Research Fellow from the Biodiversity Research Center, Academia Sinica in Taipei, Taiwan. And today I'm going to share with you some of the new latest result that we have been working for about 3 or 4 years. That would be the seawater temperature regulating the mesopelagic fish community over the past 460,000 years. Here, let me just use the pointer. Yes. So here's the link to the paper that we just published some month ago. This is in collaboration with 3 PIs from the National Taiwan University, Ziling Wei, Ziling Ho, and Li Lo. So before we get to this topic today, I would like to spend some of well, just 1 or 2 minutes to just quickly introduce myself, as I'm pretty new to this community. So I'm a marine paleontologist. I mainly work on fish fossils. The material I'm using is called fish otolith, which we will talk about that shortly. But my team is working on various marine organisms and their fossil record. So here we have two main goals. One is to describe the biodiversity hotspot from the West Pacific using their fossil record. So this is a very traditional paleontological work. The other one is to use some environmental proxies to understand how geological events have affected all these communities. For example, are there any nature? What are the nature and human impact to the ecosystem, and what we can learn from the fossils or the subfossils in the sediment. So that bring us to today's topic. What are the nature disturbances to the marine ecosystem, especially the deep sea, to the mesopelagic fish community here? As many of you are aware that mesopelagic fish community is so important to functioning properly to maintain the function of the ecosystem in the deep sea. They are both prey and predators, and they are very important in transporting all this nutrient from the surface to the deep sea, and to maintain also the trophic levels. But when it comes to nature disturbances over a longer period of time, for example, the geological timescale, then we really don't know what is the responses to this kind of disturbances. For example, we know that the climate and oceanography in Quaternary is governed or regulated by a series of glacial and interglacial cyclicity. Is this kind of cyclicity affecting all these mesopelagic fish community? That would be a very important question, and also a very interesting question to explore. So to answer, we would like to answer some questions related to this kind of disturbances and responses from the this mesopelagic fish community. To do that, we need a very continuous fossil record and also a high resolution one. One of the methods to approach this question will be to obtain the deep sea core. So we're going to study the ODP core from the Solomon Sea, which is situated just north to Australia. Here's the ODP site. The core is called 1115B, and actually this core is a very long one. You can go back to 3 million years ago, but we are going to analyze the core top, which extend back to only 460 kilo years. So it's just a very short part of the core. And we choose this core because, well, firstly, because we are very familiar with that. We have a very nice age model well established over I think it's 2 decades of study. And so we have a very good age model. And secondly, this core is composed by mainly the high content of biogenic carbonate, full of fossils, full of forams, full of other organisms. So it might have a chance that we can find some fish fossils there. And finally, because this is located in the Solomon Sea, which we consider as the edge of Western Whampoa, the Pacific Whampoa. As you know, that Pacific Whampoa is a very interesting place because we have SST, sea surface temperature, annual sea surface temperature above 28 degrees Celsius, which is so hot. That might give us a reference point because we know in this future projections that we will have a higher SST. So that would be a very good starting point. So we actually found many fish fossils. So the fish fossils, you can think about that as there are several types. Some teeth, vertebrate, spine, scale, and fish otoliths. So I'm going to briefly introduce you what exactly fish otoliths are. So fish otoliths, you can here refers to calcium carbonate crystals that you can find in every teleost fish, the bony fish, in the ear organ. Here's the brain, and just beneath the brain, you can find this inner ear organ, and inside that, you find 3 pairs of calcium carbonate crystal we call fish otolith, and one of the pairs, sagittal or sagittal otolith, is our main focus for research. The physiological function for the otolith is to help hearing and to maintain the balance in the water for fish. What's so special about fish otolith is that the morphology of the otolith is actually taxon specific. I'm not saying that these are species specific because sometimes it's really hard to use the morphological data to identify the otolith to species level, but for family or genus level, I think it will be pretty solid. And I myself is a otolith specialist. I've been working on identifying all these otoliths for like 10 or 15 years. And so identifying otoliths to family level is quite easy. And so what we find from the core, actually we extracted more than 1,000 otolith specimens from a series of 343 sediment subsamples. So you have the core, and then you subsample all these this mud and sieve it, and otoliths are there. And this is very interesting because usually when we study deep sea cores, those people who study deep sea core usually focus on forams, and they just ignore all these fish fossils, including otoliths and teeth. But they are there. I'm sure that when we examine more cores on deep sea, there will be more data from fish. Okay, The coverage for the taxonomic coverage is 28 taxa belonging to 12 families. I think we got a very good picture of the whole mesopelagic fish community. So let's just take a look at what we have for the raw data. Here in the right-hand corner, we calculate otolith accumulation rate for all these otoliths by multiplying the number of otolith count with the linear sedimentation rate and bulk density of the sediment, and we get this what we call OAR, otolith accumulation rate, that represent a specific time in the past at a specific site, which we extract the core, and that how the mesopelagic fish community look like at this time and space. Okay, so from the raw data, visual inspection, you start to see some pattern. So this shaded area indicate the interglacial stage. The white area indicate glacial stage. And you see that, okay, the OAR seems to fluctuate and correlate well with glacial interglacial stage. So this is the raw data. If we try to, we will manipulate all this data later, but first let's check what are the dominate taxa or taxon, which controls the whole data. Firstly, this is and in this upper panel, you see this total otolith count, and the lower panel is the OAR represented only by Myctophidae, the family Myctophidae. You see, there's not much change, so it is actually regulated as a whole by the Myctophidae. And we're gonna talk about the data in more details. Firstly, we did a standard smoothing method, which we called moving window method. For time series data, we usually use that. This is called a 10K moving window method. Okay, so we smooth the raw data to see if there are any changes for the pattern. And two descriptors, one is the OAR, of course, that represents fish production, or you can think about that as a fish abundance. And the other one, because we are able to identify the otolith, so we'll be able to calculate the fish diversity, of course. We use here numbers to indicate the fish diversity. This is how it looks like after the smoothing. So it's very, very clear that during the interglacial stages, MIS 1, 5, 7, 9, 11, that we have all these interglacial stages. MIs represent marine isotope stage, that we have a kind of higher OAR. Look, it's higher compared to the glacial stages. And what about the fish diversity? Well, before that, we run this is called a single spectrum analysis. This is just trying to see the cyclicity for the time series data. You can see that this line represents the significance, and here at 0.01 frequency, which represent 100K cyclicity, there's a significance here. So that means glacial interglacial stage, because in the late quaternary, the glacial and interglacial stages, the frequency is about 100K. Okay, so what about the here number? So we actually see the opposite trend. For example, here, we have relatively higher fish diversity during the glacial stage compared to the interglacial stage. Okay, so to summarize, the first finding here. During the glacial stages, we have lower abundance but higher diversity, while during the interglacial stages, we have the opposite trend. If we have data with this is the relationship between the OAR against the hill numbers, we have this kind of trend that increasing OAR will have lower hill numbers, lower fish diversity, of course. So what could be the driver driving this pattern? Here, we pick up two very important drivers. One is productivity, one is sea surface temperature, of course. First, let's look at the sea surface temperature. We got a set of data from a nearby core, which is about 10 kilometers away from our ODP core. And we have a very good data from that core for SST. And you see that SST is very clear for glacial and interglacial stages. And here I just want to correlate with our OER data, and we think that it's correlated pretty well. On the other hand, if we want to correlate with productivity, for example, these are the data from our core, or TOC. TOC means the total organic carbon mass accumulation rate. This is a proxy or indicator for the productivity. You see that there's no really a good correlation with our OER core or record. SAR is another proxy, sand accumulation rate. Remember that our core is composed mostly by biogenic origin. So core sand will represent all this productivity or the primary production. So it doesn't correlate well. And we think that probably it should be the seawater temperature regulating all this pattern. Here, there are two points that it's quite important here. So in this Solomon Sea, 26 degrees Celsius is very, very important because this is the boundary separating the interglacial stage and glacial stage. Look, above that, we have interglacial stage, but below that, we have glacial stage. There are also two very important and very crucial time that you see that we have SST above what we have today, okay? So this is called MIS 5E and MIS 11C. These two time interval is very, very, well, I would say many research has been done with these two time interval because this will be a very good analog to the warmer future. So we will see what happens to this fish community. But first, let's talk about how the temperature can regulate the fish community. So if we plot the SST with both OAR and the hill numbers, you see a hump shape gradient, temperature gradient. So look, so these are indicated with the blue line here. And the peak of this hump shape is a bit different for hill numbers and OAR. For example, here, the peak of hill number is around 26 degrees, whereas for OAR, fish abundance is a bit higher. So when it comes to the glacial stage, which is below 26, you have relatively higher fish diversity, but the abundance is still quite low. Okay, when it changes this setting to the glacial to the interglacial stage, sorry, then it's about 26 to about 28 degrees Celsius. You have the highest number of fish abundance, but lower fish diversity. That's what we found. There will be a 1.5 to 2 degrees Celsius difference to regulate all this community in terms of but abundance and diversity. We are very interested to know what would be the possible scenario for the warmer than present oceans. So we will be looking at these two, MIS 5E period and 11C period. Look what we found. Apparently we have, the decline of fish abundance. And what about the diversity? Because you expect when the abundance is lower, then diversity will be higher, right? But this is not the case for this extreme event. You find also a decline of fish diversity. That may indicate if we are facing the warmer than present ocean, if we are seeing something that 2 or 3 degrees higher than 28 degrees, you will have lower abundance, lower diversity in the future. So who will be there? So that will be a very interesting question that we would like to show you. We did MDS, multi-dimension scaling, to indicate that who will be the dominant species or fish that can be found in the glacial stages. It turns out that this Sternotherkidae, hatchet fish, will be the dominant species during the glacial stages or the lower temperature. Okay, what about the extreme event? Well, the Mectophidae is always the dominant for our fossil record, but the Bergamessorortidae, the codlet fish, will be our future. Okay, so probably if the SST, the seawater temperature continues to rise, then we will no longer see many of these Sternotherkidae, but many dominated by Mectophidae and the Bergamessorortidae. This is the most resilient taxa in the warmer than present oceans. Okay, this is the final slide that I would like to summarize what we already found. Okay, so during the glacial stage, which is around 26 Celsius in this area, we have a highly diverse fish community, but at the same time, we have a lower fish abundance compared to the interglacial stage, which is about 1.5 to 2 degrees higher. Actually, this is quite reasonable because if you have higher diversity, you spend a lot of energy in predator-prey interactions and competing for the resources, so you will have a lower abundance. And whereas in the interglacial stages, the temperature is warmer and fewer fish can be adapted to this kind of environment, and they will be able to propagate to large numbers, and that's what we found. When it goes higher to a tipping point above 28 degrees, then you will encounter something like this. It's pretty sad, but we only have few species of fish and lower number of the fish abundance. Okay, I think that's it. For that, that's all I have to say. Thank you very much.
Thank you so much. That was great. Yeah, awesome. What a good study, and really especially interesting to look at the past, basically, to look at the future, which is, yeah. It's always the question like how climate change or warming in general affects mesopelagic fish, and yeah, it's pretty cool to see that. Any questions, anybody?
Question. Really cool talk. Thank you for sharing all of that.
Thank you.
I noticed that your analysis has mesopelagic fish and epipelagic fish included, right? Or sorry, yeah. those two groups. And I'm wondering if your analysis shows any interplay between periods when the epipelagic might be really, you might have high fish production in the epipelagic but lower in the mesopelagic, or the opposite under certain climate
That's a very
conditions.
good question. Actually, during the first round of the analysis, our data shows that there are some opposite trend for the epipelagic fish and mesopelagic fish. But the problem for the detailing all this is that the epipelagic fish, they don't left really a huge number of otoliths in the record, so it's not really I would say the data is not so strong, but we find some evidence for the opposite trend.
Okay, cool. Thank you.
Yeah.
Why do you think they don't leave as many otoliths? Just because they're a different species, or is it just they dissolve on the way down or something, or what's the?
Yeah, that's possible. Because during the transportation from the surface to the deep sea, because the core is actually from about 1,300 meters depth, so it's quite deep. I think we'll get some data is lost during the process. Yeah.
Mm-hmm. Interesting. Yeah. Okay. Anybody has any question?
Yeah, I've got a quick question for you.
Yeah.
Have you been able to look at the differences in the size distribution of your otoliths over time to look at periods in which the populations might have been doing particularly well based on just their sizes?
Yeah, so that could be our next project. We didn't measure the size of the otolith yet, but because the size will have a lot to say, and I think there will be another story. Yeah, thank you.
Thanks. If you don't mind, I have another question. So it looks like you're proposing that temperature is kind of one of the correlates for this kind of change in community. Can you propose perhaps a mechanism in which temperature is exerting this change in either diversity or abundances, such as, I don't know, have you thought of ideas about how latitudinal variation during glacial and interglacial periods might be driving species geographically to certain areas?
Yeah, so that's one of the questions that we have been criticized, because we only have one core being analyzed. If we got, for example, several cores from across a larger geographical area, that would be possible to say if that's really the temperature driving all this or not, because I think it's not necessary that temperature can drive all the mesopelagic fish community in a whole area, because there's just regional situations. For example, the oceanographic current can change a lot of things. They are part of the region that is very relying on this upwelling system. I think that would be a very totally different story. But at least from this area, on Solomon Sea, that we find the temperature could be one of the major drivers here. Yeah.
I think it's super interesting that, you know, you guys are working with these IODP cores are so fascinating. It's a single core that takes, you know, obviously lots of multidisciplinary work to get there, but you're able to say a lot with one core, and I think that's really cool. So, you know, I think it's clear that in the future, the more things that we can identify in these sediments, and we can look at more and more cores, and thankfully there's institutes all around the world that have done a great job in terms of preserving those sediments. So the record's there. It's cool.
Thank you. Yeah.
It's an interesting talk. I just have a quick question. I'm wondering, how is the smallest taxonomy that you can identify from the fish otolith fossil? Just like a genus, or to the family, or even to the species?
Okay. So firstly, we need to screen the smallest otolith, I guess. We do a mesh size 500 micron. So we believe that below that, it's just coming from the juveniles and all these very, very small fish that we are not going to spend much time on identifying all these juveniles because nobody will believe you. And the otolith larger than that, we will be able to say, okay, so these probably are coming from sub-adult or adult fish. And that for those kind of individual that the otolith is quite, well, I would say is more stable. The morphology of the otolith, it becomes stable. And that can be easily identified to at least family level. I'm able to do that quite quickly because there are not many families in the deep sea, and compared to the coral reef, of course. But, in some cases, we are able to go down to the genus or species level. It's because that we have a very good reference comparative material in our lab. We got more than 25,000 otoliths to compare, which is quite large, I guess. So you really need a very good reference collections to do that.
Yeah, thank you.
Thank you.
Awesome. Thank you so much. And thanks again for both our speakers delivering such a great talks. If you have more questions, please feel free to send them an email. I think that's all we have time for today. But if you want to join the next JETZON call, just sign up to our email list. You only have to sign up once, and then you get just updates and the Zoom link sent. So yeah, feel free to join our next calls. And thanks again for amazing. I learned so much today, so I love it. Thank you. See you next time.
Thank you, Svenja.
Mm.